Okay, now we will go a little bit further on decomposition process. So this is a key process in the soil that is essential for the ecosystem. So the bulk of terrestrial net primary production, or NPP, along with the bodies and excretions of animals, is returned to the soil as a dead organic matter. Some 90% of terrestrial net primary production eventually enters the soil system through dead plants in grasslands, through leaves, roots, and woods in the forest, and through organic residue in agricultural fields. The decomposition subsystems serves to reduce dead residues to return back to atmosphere as carbon dioxide and also as soil organic matter, and to release nutrient elements for entry into soil food webs and ultimately for reaccumulating by plants. So the decomposition process drives complex below ground food webs in which chemical forms of nutrient elements become modified. So it is responsible for the creation of long and short-lived organic compounds important in nutrient dynamic, and it fuels the formation of soil structure. So terrestrial plant growth highly dependent on the decomposition system, particularly in oligotrophic soil where nutrient stocks are held in litter and soil organic matter rather than in mineral soil. So at the same time, heterotrophic organisms in the soil are ultimately responsible for ensuring the availability of nutrients for primary production. So thus, the two subsystems, which is the primary production and decomposition, dependent upon each other. So how decomposition works? So basically, the decomposition per se is the catabolism of organic compounds in plant litter or other organic detritus. As such, decomposition is mainly the results of microbial activities. However, the term decomposition is often used more generally to refer to the breakdown or disappearance of organic litter. In that context, the decomposition of organic residue involves the activities of a variety of soil biota, including both microbes and fauna which interact together. So the term uh, litter breakdown has been applied to the interactive process which results in the disappearance of organic litter. So if we want to see the influence, the rates of litter breakdown, some integrating variables need to be considered. Again, litter breakdown rates are the results of the combined activities of the soil biota. Breakdown rates may be used to evaluate effects of disturbance on the entire system. So for example, uh, here conversion of agricultural system into conservation tillage system will affect soil biology. So notably, by shifting the composition of microbial communities and increasing earthworm population densities, but with changes in other soil biota as well. So we can evaluate the consequences of these changes in soil biota by measuring litter breakdown rates. So other integrating variables including soil respiration, formation of soil structure, and also nutrient dynamics. So we can measure these variables and all are important for ecosystem function. So soil respiration estimates biological activity generally and is dominated by the microbes. So soil structure is the result of combined actions of biota and climate on mineral substrates. So we will discuss further in a nutrient cycles. So please note that nutrient dynamic are the most valuable of the integrating variables for predicting primary productivity. So although microbes are responsible for the biochemical degradation of organic litter, fauna also are similarly important in conditioning the litter and aiding in microbial actions. So the combination of microbial and faunal activities results in a set of positive interactions of the type term facilitation. Okay, so results of the interactions are likely to be more significant than by either components, animal or microbial, if they're acting alone. So they need to work together between the two. So therefore, litter breakdowns rates vary between and among ecosystems and localized and broad geographic scales. So as functions as soil biota, substrate quality, microclimate, and also ecosystem condition. So litter bags, okay, have been a valuable tool for comparative studies of rates for this litter breakdown. See, so basically, so the litter bag usually use the mesh bags with the holes, usually using nylon. So they put a leaves inside in the this mesh bag, this bag containing a known mass of leaf litter, and then after that they put back in the forest floor or any land, so to look at to replicate the time of the leaf drop. So litter bags are then collected on a timely schedule, and the remaining mass, so after a certain period of time, will be measured. So of course there is some problems using this litter bag technique. So for instance, one of the problem is if you use large meshes or you have a larger holes, this will allow some leaf fragments to escape the bags. So thus this will overestimating the decomposition. But if it's too fine, so you use very small, very fine mesh bags, so it can also exclude most macrofauna that, again, remember, that can facilitate the process of the decomposition. Yet this will problem. So which means that the macrofauna, because the hole is too small, they cannot enter the bag and facilitate the decomposition. Yet the problem here, when you use too fine mesh bags, it can underestimate the decomposition rates. So now go and do some readings and try to answer these questions. So first, which ecosystem or environment or condition has faster decomposition rate between temperate and tropical forests, between agriculture and the forest, between tillage and non-tillage systems, between buried and surface litter? Okay, so yeah, no worry, I'll give you the answer. Okay, but can you explain to me why? So we already discussed about the roles or the association of soil fauna with litter decomposition. So the upshot of these experimental manipulations has been to demonstrate, okay, the graph here, try to demonstrate that bacterial, fungal, and faunal members of the soil biota all have significant effects on the litter breakdown. You see here, without fungicide, okay, when you look at this graph, without the fungicide, all this dot is with when they use the fungicide, okay. So without the fungicide, bactericide, and arthropod repellent, okay, without this fungicide, bactericide or arthropod repellent, the decomposition rate is significantly higher. So given that actual breakdown and decomposition rates are a function of the interaction among the various biota and with substrate quality and climate, the rate estimates derived from manipulation must be accepted with caution. So the main effect, however, seem very very clear, which is there is a the relationship or the roles played by the fungus, microbes, bacteria, yeah, fungus, microbe, and also the soil fauna. So we look into the carbon cycle and nutrient movement. Into the carbon cycle, some nutrient cycles, and also nutrient movement on the next class.